Welcome to the April 20th VanBug seminar, the last seminar of our 2022-2023 season. Today, I'd just like to remind you all to keep your phones silent. This event is recorded, so if you have any qualms about that and speaking, just be aware that this is recorded, and it will be uploaded later to YouTube on our YouTube page. So today we're going to have a trainee talk by Hans Getze, and it's going to be followed by our featured talk with Yongjin Park, and then we'll have our normal food and drinks happening after. I'd like to take a moment to acknowledge the VanBug Organizing Committee. We're led by Dr. William Shao and Amy Lee and Faraz Haq, and we have a wonderful group of volunteers spanning SFU, UBC, and Langara College. We have an upcoming event. We have the late terms talk competition. Submissions for that are now closed, but you are still encouraged to register to attend the event. There will be meals. There will be a lottery at the actual event for participants and for the audience alike. So here are the rules of the talk, so you know what you'll be watching or getting into if you have submitted already. So there are going to be 3-minute presentations in accessible lay terms, generally from just bioinformatics and bioinformatics related projects. Please contact us if you have not been confirmed, your registration has not been confirmed. And again, remember to RSVP if you plan on attending as just an attendant. We have a $30 gift card for 2 lucky attendees as our raffle prize. And then, as this is the last session of our season, we'd like to just say that we have a Suggest the Speaker button on our vanbug.org website. We take those suggestions into account when we plan our season. So if there's anyone that you would like to see, please suggest a speaker, and we will keep them in the consideration for next year. And our sponsors are the Graduate Studies in Bioinformatics at UBC and SFU, Genome BC, Imagia Connexia Health, Stem Cell Technologies, Langara College, St. Paul's Hospital, and we are affiliated with 2 other bug organizations, Monbug and Torbug in Montreal and Toronto, respectively, and we are also affiliated with the International Society for Computational Biology, ISCB. Okay, so this is Yongjin's talk title entitled Learning Deep Biology with Shallow Statistical Models. But before that, we have our student talk by Hans Getze, who's a PhD student in Carolina Trapini's lab at UBC, and he's going to be talking about PuPPy, an automated pipeline to design taxon-specific primers in microbial communities. So without further ado, we'll have Hans come up. Yeah, this is the microphone that works. So you plug in here. And we'll just check.
Will you be monitoring Zoom for questions later?
Yes.
Perfect. So I don't have to.
You don't have to do any of that now. Okay. And then you just go on full screen, and then you are coming in through here.
Yeah.
Yep. So
perfect. All right.
Great.
All right. Welcome, everybody. Thanks for coming out tonight. As introduced, I'm Hans. I'm a PhD student in Trapini Lab. And in my project over the last year, we've been developing a computational pipeline called PuPPy. It's a little bit of a different title than before, but it performs the exact same function. It designs taxon-specific primers to detect and quantify microbes in any microbial community. When I started working on this pipeline, we kept in mind the gut microbiome, which is the focus of our lab. That is because the gut microbiome is an incredibly complex ecosystem where there is a huge biodiversity along the gastrointestinal tract. This huge biodiversity is parallel to a diversity in physical environments. For example, parameters such as pH, osmolality, and temperature. They all vary across different timescales that can range from minutes, years, days, and they can actually have a huge impact on both the gut microbiome and human health and disease. That is because it's really important for us to study why these parameters impact the gut microbiome and how. Now, in our field, some of the most common approaches that we take to study the impact on gut microbiome are generally in vitro studies, but most importantly, in vivo investigations. These are commonly involve mice, either germ-free or already pre-colonized, and some kind of sequencing. Two of the most common approaches are 16S rRNA sequencing, which focus on just that one particular gene, or metagenomics, which investigates the entire genomic pool in the sample. Now, while both approaches are great to characterize and investigate microbial communities to determine their composition or their relative abundance, they both have limitations. In particular, 16S has one of the main limitations that they have low taxonomic resolution. That is, we often cannot get beyond the genus level. And it is database dependent. That is, if we don't have a particular reference 16S gene prior to the sequencing and the analysis, we will not be able to identify those organisms in the sample. And also, another main limitation is the copy number variations that are specific to each organism. The microbes have different numbers of 16S copies within a single genome. And thus, when we look at 16S to get relative abundance, we often cannot get proper estimates. And again, one other limitation is relative abundance. We would like ideally absolute copies, but we can only get relative abundance. Similarly, metagenomics also has limitations. One in particular is that it often requires high sequencing depth. That is because low abundant organisms require more reads to be able to be detected in a sample, leading to higher costs and more complex, more computational challenges due to the intricate nature of the data set. Again, like 16S rRNA sequencing, we have the same problem of relative abundance as opposed to absolute genomic copies. Now, when considering these approaches to study gut microbiomes, we see why we could use another approach to, one, decrease the cost of the sequencing in our experiment, but also increase the resolution to achieve really high as possible, for example, up to the strain or substrain level, while also achieving absolute quantification. Now, to this goal, we developed PUPI. PUPI stands for Phylogenetically Unique Primers in Python. This is a fully automated pipeline that designs taxon-specific primers in any microbial community. When I mean with microbial communities, I don't just mean bacterial communities, but also viral, fungal. As long as you can provide the input files, PUPI will be able to work and design taxon-specific primers. But let's dive into the details. PUPI essentially finds unique genes within a community and designs microbe-specific primers. These are primers that selectively amplify individual microbes within that community so that you don't get unspecific or unintended amplification. Similarly, we can find group genes, which will be used to design group-specific primers that selectively amplify user-determined groups of microbes. For example, just the 3 bacteria out of the entire microbial community. In terms of the computational workflow, as I mentioned before, as long as you can provide the input, it will work. In our case, the input are coding sequence files. So any gene files that you can input into the pipeline will be accepted. And then the pipeline works in 2 key steps. The first one is PUPI align, which performs many against many local and pairwise sequence alignment. So technically it's pairwise, but it's many against many. That is, we compare each gene in the community against all the other genes present. And we repeat this for all the genes and ultimately get this blue here alignment file. The second step is PUPI primers, which designs taxon-specific primers based on any user-determined parameters, specifically regarding the primers. You know, you can change length, annealing temperature, and all these parameters. PUPI primers takes 2 inputs. The first one is the blue file, the alignment file, and also the target CDS files for which you want to design primers. Here, it branches into 2 possible options. It can either design unique primers where we identify the unique genes, then we use primer 3 to design microbe-specific primers, or we identify group genes, and then we use primer 3 to design group-specific primers. Just as a warning, we always recommend testing primers in the lab, because as perfect as they can be in silico, they will not necessarily work in vitro. Now, some of the applications that we've already identified and used so far are for the detection of microbes in defined microbial communities by PCR, but also absolute quantification in microbial communities by digital droplet PCR or qPCR. I noticed how I mentioned defined. It can also be used to detect and quantify microbes in undefined microbial communities, for example, from mouse fecal samples or patients where we don't know what we're starting with. We have actually a couple more ideas in mind, but right now we've only validated, or we are validating, these two detection and quantification functionalities. Now, as I just mentioned, today we're only going to be talking about the detection and quantification aspects. For the detection, we designed 3 increasingly complex microbial communities to test the limits of the pipeline. The first one, we'll call it the PEG community, and it's 10 phylogenetically distinct microbes. The second is a species strain substrain called SSS, and here we are increasing the complexity by having more related microbes to test the specificity of puppy. And lastly, we're working with a complex or undefined microbial community from mouse samples. For the quantification aspect, we're taking the PEG and SSS communities, and we're trying to see how good our primers are to quantify each individual microbe in the community. Now, focusing on the PEG community, this is a list of microbes. It's not that important, if not just focusing on the average nucleotide identity plot, the ANI. Here, what you want to see is the larger the circle and the more red, so the darker the red, means that the 2 genomes are more related, they're more similar. And as you can see, they're not they're very different, except Biobatis, which is a Bacteroides vitis, and B theta iota omicron, because they belong to the Bacteroides genus, so they're more related. For each of these 10 microbes, we designed microbe-specific primers, and we validated them in vitro with PCR. Here I'm showing an example of a validation that we do for each microbe-specific primer. We first of all test that primer pair against the gDNA of the intended target, just that gDNA. We then run the entire 10-member pool with those primers. We then remove the intended target to check for unspecific amplification against the remaining members. And then we have a no template control. Now, this is a binary heat map where we see in black the amplification, successful, or no amplification. And as you can see, each microbe-specific primer selectively amplified the intended target organism and nothing else. This was exciting, but it was a phylogenetically distinct pool of organisms, so not that surprising. So we moved on to the more complex community, which is the SSS. Here we chose 3 enterocluster species, 3 B theta iota omicron strains, and 4 E. coli K12 substrains. And as you can see from the ANI plot, we confirm that they are indeed increasingly related, where the species are there's the lowest genome similarity up to the substrain, where they are clearly very, very similar. For this community, we designed, again, microbe-specific primers for each member, and we validated the exact same way as I mentioned before. And we were really excited to see that it selectively amplified only the intended organism, but nothing else, even at the substrain level, where there is incredibly high genome similarity. Now, just to pay attention on the second last row, which is E. coli MC14100, it's blank, because when we run the pipeline, we could actually not identify a single unique gene for that organism, so we did not design any primers. This is also future directions. I'm thinking about ways to design unique primers, microspecific primers for non-unique genes, but future directions. Focusing on the primers, we've also designed group-specific primers. Group-specific primers for each taxon that selectively amplify each taxon. And for the validation, we ran the gDNAs of the specific taxon of interest, then the 10-member, the pool without that taxon for the specific primers to check for unspecific or intended binding, and then the no template control. And going again to the plot, we were super excited to see that even the group primers were specific to their own taxon and didn't amplify anything else, suggesting that PAPI design primers, whether they are microspecific or group-specific, they selectively amplify the intended organisms independently of microbial the composition complexity, or the composition itself. Now, we are currently working on a complex and undefined microbial community. It's a bit of a spoiler, we won't see the results, but we were able to actually selectively detect our intended microbes with microbe-specific primers, even without knowing who is there in the community. So this was really exciting. Now, the last part of the talk is about the quantification aspect. As I mentioned before, we're focusing on the PEG and SSS communities. What we did here is we grew each organism in their own preferred media, we extracted DNA, and then we pooled known amounts of genomic copies for each microorganism and sent the pool sample for 16S high throughput sequencing, metagenomics, and then we're currently doing ddPCR, digital droplet PCR, using our microbe-specific primers. The goal here is to quantify, get absolute quantification with our primers, and evaluate how it does compared to the two most common methods that are 16S and metagenomics. Just as a spoiler for part of the results that we're collecting right now, we're still working on this. For the PEG community, you see that metagenomics, sorry, it's really tiny, I had to add it last minute. Metagenomics does okay, not perfect, but it does okay at quantifying the relative abundance of each organism compared to the in vitro sample, which is the sort of the gold standard here. But kind of expected, but it's still, every time I see this plot, it's pretty shocking. 16S really does not do well at all at quantifying bacterial abundance. As you can see in green, the Bacteroides become incredibly overrepresented compared to the in vitro pooled amounts. And that's probably because of gene copy numbers, which really emphasizes that 16S should not be used for abundance quantification unless it's necessary. And yeah, just a few key takeaways that I hope you leave the talk with. PUPPY is a fully automated pipeline to design taxon-specific primers without the need of memory-intensive databases. And these primers designed, whether microbe-specific or group-specific, can be used to selectively amplify intended targets independently of the community composition or complexity. And lastly, PUPPY may represent an inexpensive, rapid, and efficient way to characterize microbial communities at extremely high resolution. So just to wrap it up, I want to say thank you to everyone, the organizers, everybody that is here, everybody in the Tropini lab, especially Michelle Fan. She's been working with me on this project, and the Zeals lab. And thank you for listening. If you have any questions, I'll be happy to take them.
Okay, we got time for one or two questions for Hans. Anyone have any questions? Got one in the back, Andrew. Okay, if you could push the little tag in front of you, it should glow red. Yeah, just put your finger on the plastic part. Turns red.
Oh yeah. Okay. So now, can I ask the question?
Yeah, go ahead if it's not turning on.
Okay. So thank you for the presentation. Really good and really nice slides. I liked it a lot. I was wondering, like, how computational demanding is the alignment step? Just because I would expect that labs would want to use the tool, but then they might not have like a big computing capacity. So
yeah, no, thanks for the questions. Great, very important actually. We are using a tool called MM62. I don't know if Eddie's heard it, and it's incredibly fast at aligning genes. We can actually finish the alignment step using Sockeye. For, I think the fastest we've run it was against the largest community was about 160 bacteria, so CDS files, and it was done in, I think, less than 15 minutes on Sockeye. I ran it on my computer and it took about an hour, I think, but it's just on my personal computer. We've run it against 400 bacteria as well, and it's really fast. It's not computational demanding. I don't think that that's ever going to be the limiting step of the pipeline.
Great.
Yeah.
And one more quick question. So that's great. So I understand that you will evaluate the performance of the quantification with a 16S assay, right? Comparing with a 16S assay, right? But you also showed that the 16S was not like really good. So is the community that you selected for this test like a good case for 16S, or or is 16? Yeah, because I'm wondering about that.
Yeah, that's a good question. We are doing 16S on both communities. We're doing metagenomics on both communities. So we're trying to be independent of community composition. So we have one that is phylogenetically distinct and one that is more related. As I mentioned before, the more related one, 16S, is going to struggle a lot because we are already taking a fragment of the 16S that is only about 300 base pairs when we do high throughput sequencing, which reduces the hypervariable regions in the 16S to differentiate bacteria. So it is a case scenario because in any complex community, we will have different strains. We will have different species. And ideally, we would want to be able to distinguish that. But practically right now, or at any point in the future, it might be impossible with the 16S because 16S can either be the same across similar taxon levels, like two species could have same 16S, or they could even change across a single genome, can have 3 copies, and they can look different within the same genome. So 16S has already its own challenges.
So a quick follow-up on that. So what would be a great gold standard for you to compare, let's say, the performance of the quantification of PAPI to a gold standard quantification? Would you compare it in the future to a metagenomics assay, or?
We're doing that as well right now. We are so right here. I only had to show you this. I'm sorry. Yeah, it's right here. I had to change this last minute, and it's really tiny.
Yeah. That's okay.
But we actually have metagenomics for the PEG community and the SSS, and we have 16S for both communities as well. I just didn't really have time because we collected this data.
No worries.
These
Thank
days.
you. Yeah,
but we're doing both for both communities.
That's great. Thank you.
Yeah.
I have a question from the Zoom chat. I'm just going to come up to the mic to speak.
Yeah.
From William Hassau, how does PUPI take horizontal gene transfer into account?
That's the problem that we are not really going to be able to account for because it's going to depend on each community. So we are just going to assume that the gene you're finding are highly conserved to that taxon or that organism. Because when you think about a community of 160 bacteria, if you need to find a gene that is exclusively present in one, it probably means that it's highly conserved. And so we're just going to assume that the chance of horizontal gene transfer may be lower because that gene is so highly conserved for that microorganism. And probably the same going to be for group genes. If you're finding one gene that is only present in that specific taxon out of 160 microorganisms, it's probably chances of horizontal gene transfer might be lower.
Well, thank you, Hans. Without further ado, we're going to move on to our featured speaker, Yongjin Park. If you want to come in and plug in, I'll just introduce you. Yeah. So we have all of them. Yeah, you don't need it. Power is, yeah, so I don't care. Then you have you right here. Goodness. All right. Then yep, there we are. Okay, great. So Dr. Park is an assistant professor in the Department of Pathology and Statistics at UBC since last 2 falls ago now. And he's also a scientist at BC Cancer. So generally what he works on is Bayesian statistics, but he's also interested in developing causal inference algorithms. Yep, he's worked with statistical genetics, regulatory genomics, epigenomics, all of the different omics. He received his PhD in biomedical engineering at Johns Hopkins, being advised by Joel Bader, his master's in computational biology at Carnegie Mellon University under Russell Schwartz, and his bachelor's in biology and computer science and engineering at Seoul National University. So without further ado, I'll let Yongjin talk.
Okay. Thank you. Thank you so much. Thanks for having me. Thanks for the wonderful introduction. I I was actually thinking, what would be the good topic to discuss in the Bamberg meeting. So I just had everything in. I have 190 slides, so I'm not going to go over everything, but I'll just see. Yeah, today's topic is more of somewhat philosophical. It's about my experience. I named it as a learning some deep biology using shallow sort of probabilistic models. So before we do, I have an acknowledgement. So I live and work and study in the traditional ancestral and ceded territory of the Musqueam people, which is UBC campus. I got fundings from NSERC and also BC Cancer and Provincial Health Authority. So before I start the talk, I just added this in the last minute after I had a conversation with students. Yeah, who am I? Basically, I'm a modeler. So how I started my sort of modeling sort of career is primarily inspired by this book. Actually, I didn't see the book when I was learning the subject because Russell was actually writing a book. And he used all the chapters in the book to teach the class. The class was incredibly intensive. I think it's one of the most intensive course in the computational biology program at CMU. Basically, but it kind of opened my eyes. At first, I didn't understand anything. So it just I was like a biology undergrad and just had a little bit of experience at CS. Russell had a deep understanding of a bunch of different methods, but he kind of sort of remind me that I should be a modeler. So basically, the book is pretty old, but I think it's a kind of a good way to understand how you could model, how you could think about the problems. And then another part of me is I'm deeply interested in science. So I also at the same time I care about the efficient and fast algorithms. So but I think the third bullet point is most important. I basically want to understand every piece of the model that I will be using or sort of developing or something like that. So yeah, today's talk, it's sort of like mostly random projection of my research, mostly done at UBC and BC Cancer. So there are different flavors of the research. I don't think I will go over entirely everything, but I'll just briefly go through some of the topics that people are most familiar with, but I'm more focusing on the Shishir Subedi's project. Yeah, the first let me start with the sort of probabilistic topic model. So I will be working on the single cell. I'll be talking about single cell data sets. So I think it's probably good to start with how the data were generated. For those who've taken my class, have seen this slides many times. So it's the single cell. I think it's a pretty interesting technology that it's beginning to change the field somehow. So now, before we had a measurement of the tissue samples, now that we can actually dissociate the samples in the mixture of cells, and then there's some droplet technology or some other type of capture technology, we can encapsulate one cell at a time by one sort of droplet, and then you run through the droplet into some sort of a nanotechnology and then microfluidics technology, and then you can actually sort of measure each of the cells going through this sort of like interesting sort of device. And then once you've done that, you can encapsulate the cells in one droplet. Then you can actually tag each cell, the whole transcriptome or genome or epigenome in the microbeads. And then once you sort of tag each cell by some unique barcode, and then you can actually mix them all and then do the sequencing. So which is kind of in a nutshell, how the single cell measurement can be done. So why do we do single cell assays, first of all? Cell is basically the fundamental unit of cell life. So we care about cellular heterogeneity. And then it can also teach us some differentiation trajectories, which is a fundamental process of biological systems. And then there's also some variation within the same cell types. That's what we have observed. And then at the same time, cellular response can actually vary from one cell to the other cell, even though they are in the same cell types, but they can have different cell states, especially in immune context. And then it can also overcome some sort of a low input quantities. So then you can be more sensitive to the actual biological signals. And the common myth is single cell data is not the cell sorted assays. So this paper actually basically demonstrate that single cell from one cell, single cell from the other cell, you see very little, mild correlation, but there's some discrepancy. But at the same time, if you put them all together, they're kind of agreeing on average. And also, it's not the same as the single cell average versus the bulking pseudo bulking data, the bulk measurements. So there's some correlations, but it's not perfect. So that's why it's worth measuring the single cell. So in the single cell world, what we are, it's a high dimensional and high volume, large number of samples. So then we will want to somehow get a feeling of what the data will look like. So in the sense, people start using some sort of a dimensionality reduction method. One example is a t-SNE, t-stochastic neighborhood embedding. Then you can project the high dimensional data for each cell onto some 2D space. Then you can actually visualize where they are. So basic idea of the t-SNE is basically it's nothing more than the optimization. So basically, you measure the probability between cell I and cell J, and which is this, and then you will come up with some Qi and Qj. That's a low dimensional probability between cell I and cell J. We want to match this Pij and Qij similarly. So how do we do that? By minimizing the distance between the probability space. So single cell now are gaining more traction in the epigenomic signals, and then one of the key breakthrough in the experimental side is ATAC-seq. So ATAC-seq was basically you can look at the chromatin accessibility, and then whenever there's open, then you can actually introduce some transposon components, and then you can chop off those sort of open regions, and then you can amplify the signal. So by PCR, by the side story, the transposon is basically first found by Barbara McClintock. So basically by looking at the patterns, the color patterns of the corn, and then you can actually see some genes it looks like it's jumping. So then that's what she found, and we're still kind of building upon the fundamental biology. So ATAC-seq is useful because it can start with the very little material, and the preparation time is significantly shorter than the previous technology like DNA-seq or FAIR-seq. But it can also it's sensitive enough to capture single cell level epigenomic data, which is the DNA accessibility. Then we can actually measure how many unique regions in a cell, what's the dimensions, all kinds of things can be answered by measuring a single cell ATAC-seq data. The technology is the same theme. Basically, it goes in the same theme. Basically, you sort of tag each cell by some sort of barcoding, and then put them all together, and then do the single cell, the sequencing, and then you can combinatorially matching. So yeah, let me skip this. So in the end, the ATAC-seq can give us some sort of features for each cell, and then for each cell feature can be mapped onto some probabilistic space. So that's why people come up with some sort of topping model. So in this case, they use latent dirichlet allocations. So I'll get to the probabilistic topping model soon. So basically, we can actually visualize all the DNA accessibility regions into some topic space, with which we can actually identify some DNA motifs of the regulatory motifs. So that's actually a pretty useful tool. So that was a kind of a quick tour of the single cell field. So now the fundamental question that I'm interested in is what is a generative model of the single cell expression data or some ATAC-seq data? So what we want to model is that gene expression matrix, which is gene by cell count matrix, and then we want to find some sort of latent factorization that's beta times H in this case. Beta is a gene by latent dictionary matrix, and then H is latent mapping, some latent dimension, and the cell. That's the common theme of this talk. So we've been using PCA quite a lot for using high dimensional data, we can actually reliably map them onto some latent space using PCA. But nowadays, people starting to use NMF, which is non-negative matrix factorization. It's a relatively classic method. So it first came up in the 1999. Compared to PCA, the non-negative matrix factorization has one strong benefit, which is you can actually tease apart the parts of, for example, this image data. You can tease apart the parts of the image, and then you can have loadings and then the dictionary. Compared to the PCA, it's a significantly different sort of property. So PCA, you can have both positive and negative values. That way we can actually mix. We don't know how to distinguish between the parts and the other parts. So fairly recently, the people got NFF sort of draw more attention in the single cell world. So this is one of the most successful story of the NMF. So by Joshua Welch, and then it's basically look at the different single cell data that can come from different species even. And then it turned out that the NMF-based sort of method is useful to integrate one data set to the other data set after some non-negative matrix factorization. You can actually plan the different type of data fairly well. Another success story of the NMF is actually International Cancer Genome Consortium. Basically, they look at all the mutation patterns, and then they treat that as a giant matrix, and then they perform the NMF, and they can actually figure out what is the mutation signature that's commonly observed across different sort of cancer genome, and then across different cancer types, and then it can actually provide some sort of more interpretable results. And what's the common pattern, like a C2A mutation versus something else? Then we can actually figure out what it is. So from that story, then nowadays NMF is kind of useful, but somehow people are not much satisfied because we've tasted the sort of deep learning. So and then the one thing is people have at first, when we first explained the t-SNE, for example, like I don't know, like 5 years ago, even like 10, people didn't understand what t-SNE means. But as soon as we start using it, and then we kind of learn that, oh, it's useful for visualizing the single cell data. And then people started using, and then NMF is and the PCA is essentially the same type of approach, but it's also the one layer. So I would call all those things as a shallow models, basically. So you have a high dimensional data, you have just one layer, the latent sort of mapping. It's pretty shallow. But now the people are trying to look at the deep models that can actually, you could inject the multiple layers between your latent dimension that you're going to interpret, and then your observation, the vector. So the estimation wise, I think the one reason why deep learning gained more traction was basically we had a significant improvement in the algorithm side. That means at first when we present this, computer science had a really hard time trying to figure out how do we actually estimate all these parameters between different layers. But they somehow realized that, oh, if you stack them up together and then take the mirror image, and then you can connect them as an autoencoder, then it's possible to just simply take the sort of like some sort of chain rules, and then you can actually just take the stochastic gradient steps. It's fairly easy to estimate. That's why people start to using it even in the biology settings. But putting all the deep learning aside, and then I'll be focusing on the NMF. The reason is because it's interpretable. What do I mean by interpretability? Is basically, since it's one layer, then we have a gene expression count data, and then which is gene by latent vector. And then each latent vector is basically a set of genes. If you could somehow find a small set of key genes for the latent vector, then we can actually estimate that. But in order to estimate this well, I think the one thing that we need is we want this to be sparse. We can't really take all the 10,000 genes then trying to understand, I think that's a pretty difficult job mentally. But if we can somehow break that down into small pieces of gene sets, then we can actually understand what that means. So that actually brought us into more sparse matrix vectorization methods. So here, this dictionary matrix, if you just estimate the, some sort of non-negative matrix vectorization type of model, then it gets very dense, especially positive and negative component can actually mix up together, which is something not really interpretable. As opposed to if you impose some sort of sparsity, then you can actually make it really sparser, and then only a couple of items are sort of lighting up per topic, then we can actually make interpretation fairly easily. So that actually motivated us, and then we designed some sort of Bayesian latent factor model. It looks like deep learning, but I have to say we only use deep learning to make the model inference easier. So we use it as an encoder part, but the decoder part is basically what's modeling is happening. It's just one layer, it's a shallow. So we will use that, and then we can actually improve the interpretation much better. So if you compare side by side sparse versus dense model, so here some parts maybe somehow corresponding to multiple topics, but here you can, as opposed to the dense model, it's heavily sort of populated. We don't know that they may cancel each other, so it's really hard to understand, but we can actually tease apart all these genes. Indeed, if you compare this side by side, it often corresponds to the known marker genes of cell types. This is a pancreas example, and then we can actually map those sort of topics onto some known cell type marker genes. As you see, it's a very much enriched one-to-one. So we didn't pose any prior knowledge, but it kind of helps us to sort of disentangle the signals. In the end, it actually improves sort of separation of cell types. So when you're dealing with the dense sort of model without any sparsity constraint, then your cell types are kind of mixed together. So cell topics are somehow convoluted, but using sparse model, we can actually nicely assign pretty much one cell type dominating per each cell. So we can actually nicely separate them. So that's how it is. So and then when you look at the t-SNE plots or UMAP plots, they are actually separable somehow, as opposed to dense model. So which is actually a big benefit. Another advantage of why we're focusing on shallow model is because you can transfer knowledge fairly easily. So if you somehow analyze all this PDAC, pancreatic ductal adenocarcinoma cells, 57 cells, and then you have some sort of cell topics. And then we want to transfer this knowledge to some other data sets. So since our model is really focusing on the genes and the topics, their genes don't change across different data sets. That's why we can actually match them. So then we can revisit some of the sort of using this single cell data, we can actually match that to the sort of bulk data. So this setup is one of the experiments, then we can actually train the single cell factorization model in this way. On the other side, we can have a sort of a TCGA or ICGC, a sort of bulk RNA-seq samples as a Y. And then we assume that the topics are somehow shared. And then we can actually estimate what is the bulk specific sort of latent vector representation, and then what is a single cell specific latent vector representation. So it's just still shallow model, but it kind of jointly trained. If you jointly train, if you look at the UMAP space, these gray dots are basically single cell. So which means that some of the red dots, which is bulk data, are actually in the same space of single cell space, but some of them are really just sort of unique. But most of them are pretty well aligned. So, and then using that, since bulk data, we don't really have a cell type definitions, but the joint analysis can give us clearly separate out all this 467 bulk samples into two groups. One is a better outcome group and the poor outcome group. And then one of the topic in here is actually, even though it's tiny, but it's statistically significant. can actually give us a significantly different survival probabilities. So if you compare two different groups in the two, and then look at this topic proportion, topic 2, then you see something different result. And another topic 4, you see, which is also another significant things. And it turned out that, not surprisingly, has to do with the immune response. So T specific cytotoxicity and the T cell apoptosis, cytokine interaction pathways are heavily enriched in this topic 2. And then topic 4, it has to do with another type of immune response, which is focal adhesion or calcium signaling or ECM receptors. They are all cell-cell interactions. They can be actually combine them together. So this is one of the good a mental exercise that we tried, and then it turned out that the shallow model has a benefit that we can do. Models are fairly easily interpretable because we can directly compare with the gene sets and then known pathways, and we can also directly integrate the different types of data sets. Like in this case, single cell data and the bulk data, we can actually mix them together. So another flavor of a data integration that we tried is in the RNA velocity. What do I mean by RNA velocity is basically we want to measure the speed of transcription, whole transcription gene regulation process. Especially we are interested in measuring the splicing steps. In this case, single cell data and also the bulk data, bulk sequencing data, we can actually separate the two types of reads, which is a spliced read and an unspliced read. And then we can measure the ratio between U and the unspliced count and the S-spliced count. Then we can separate them out at different stage of the splicing process. Then in the end, as a result, we get the two types of matrix: spliced matrix, unspliced matrix. And then we can measure the sort of the dynamic nature of transcription regulation process. So a typical approach in systems biology is we use ordinary differential equations, which requires us to sort of estimate the splicing rate and the mRNA degradation rate. So basically looking at all this phase diagram between the spliced and unspliced, we can figure out the parameters. Yeah, that's technically possible if we have a full trajectory data. So that's then we can reliably estimate all the rate parameters. Then using the velocity, we can actually map those vector field. How is the transcription is moving along in the latent space? That's great if you have a full data set. But however, in our case, in the cancer data set, normally what we found is we have a very incomplete data sets, which means if you plot the splice and unspliced as a sort of scatter plot, you see it's only one side somehow sort of activated. It's very tempting to draw the line in between here, but I found that it's a little bit not satisfying, especially looking at this some zeros and then also some other side is a kind of highly skewed to the splice or some other on the other way. But the problem is it'll be, we don't know whether actually there were some sort of steady state solutions in this gene expression patterns, spliced and unspliced patterns, like the same way for the other genes. So instead of solving really complicated ODE model, which is totally nonlinear and there's no closed form solution, if you are dealing with the many genes, one gene, yes, is possible, but many genes, no. So how do we do that? Instead of doing that, we come up with something simpler model, which is also another shallow modeling, which is we modeled the spliced and unspliced count jointly. But the splice side, we actually add some sort of another rate parameter, which we denoted by delta, which is kind of really the sort of the velocity change. So, and then there's some static loading that so rho plus delta will be the total rate of the splice count, whereas the rho is just the unspliced count. So the idea is we can really separate what is a steady state sort of topping models versus what is a dynamic topping models. So going the story faster. So I think we did that on the pancreatic data sets, pancreatic cancer data sets, and then we identified the delta topics, we call it. We get something like a 30-ish reliable topics, and then we can actually separate the cells into different components. And it turned out that those delta ratio it's very meaningful because we transfer the knowledge to the bulk data and the TCGA data. Then we can actually see some of the topics. For example, delta topic 4, it's a significantly sort of different between the two groups of patients, like the survival curve. And then delta topic 11, we also see the same level of higher level of significance. Whereas the corresponding the row topic, and then another sort of shallow model that we introduced, we didn't see much difference. Some significant, but not terribly significant. That's why so delta topic sort of captures the unique nature of the disease progression. That has to do with the RNA velocity, transcription velocity. So for the sake of visualization, we can actually project the topics onto some sort of vector field. In that case, we can actually map that some disease-specific topics that sort of increasing the survival rate that goes this way, whereas some of the poor outcome delta topics goes the other way. So we can actually visualize that the delta topics go in different directions. So and overall, we found a stronger effect size compared to the other sort of topic modeling issues. And then is it something unique? Then is it something algorithmic artifact? So we with the different topic model approach, and then we compare them at the gene level side by side, and the delta topics are somehow unique, which means it's not really captured. It's not really captured on the other side. So maybe some of them they can be. We can still recapitulate same topic patterns, but overall it's more sensitive in terms of picking some dynamic topics. So yeah, the paper is still in revision, so we need to address reviewers' comments. Okay, so that's the sort of the probabilistic topic model on the single cell data. So far we've been working, and then I think we've been doing some successful work, and there's still more to do. Yeah. Before we do that, do we have any questions? Then feel free to jump in. So yes.
Thank you so far. This is really interesting. For your topic models, are you, from what I recall from like linear distributed allocation, you set a number of topics beforehand.
Yeah.
Do you have a set number of topics in your model?
Right. So your question is, how do we set the number of topics?
Basically, yes.
Yes. I think I try to avoid that issue. There's several ways. One way we did is basically we impose some sparse parameters, sparse regularization conditions. In our case, I used Bayesian sparse spike and slap. In that case, we can somehow automatically select the topics that are sort of completely zero and that actually has some part of non-zeros. That way, I kind of, as long as your computer can process, I add a sufficient number of topics, and then maybe some of the topics ended up being empty. So in that case, I select the yeah.
Okay, I see. So basically, you said let's set your upper limit of the number of topics to be like 1,000 or something.
Right,
big,
right,
and then
right.
your sparsity requirement.
Sparsity parameter will
okay.
we'll take care of it. Yes.
Okay, that's really interesting. So one more thing, did you guys consider doing like a Dirichlet process, sort of like basically non-parametric approach?
Yeah, that's a great question. Yeah, I think that's definitely on our to-do list. But in terms of implementation, I think it's kind of challenging.
Okay.
Because it's non-parametric Bayesian is kind of slow.
Right.
Yeah.
Okay.
I mean, we could consider some variational approximation. Yeah.
Okay. Great.
Yeah.
Thank you. Appreciate it.
Yeah.
So it's like a practical number of starting topics thing.
Oh, I don't know. 7.
7.
Yeah, something like that. We actually start with the 50.
50. Okay.
Yeah. So there's some numbers like in single cell, especially single cell RNA-seq analysis. There's some numbers people tend to use, which is 500, which is a highly variable number of genes they use, and then the 50 is the number of the latent, dimensions they use. So actually, we don't need all the 50 topics, yeah. But we try to be more generous, like 50 or 100, yeah.
And also, have you compared like the output gene sets to like existing like gene signatures people use for like subset proportion estimation?
Right. Yeah, I think that's still, it's a little bit open question, but how do we really refine that? For example, some of the gene sets in one study may not hold up on the other study. So in that case, what people have done, I think one of the successful approach Dana Pierce group have been using was they actually really include the gene sets in the model, and then let the model somehow sort of prune out some of the irrelevant ones. So I think that's a reasonable approach. But yeah, we didn't try that. Yeah.
Do you have any questions on
Okay. Can I know?
Sure.
Are you planning to integrate the single cell
ATAC-seq? Yeah, I think that's a great question. Yes, that would be great, but it's computationally more expensive. The reason is we were previously RNA-seq, we were talking about something like 10,000, 20,000 features. Now ATAC-seq, we have to somehow up to 1 million features, and the million cells. I think it is a kind of a challenging computational problem. And then we will try to come up with some solutions. Hopefully I can address that a little bit. Okay. So another sidetrack is also the topic modeling thing, but it's we were talking about something slightly different problems, which is a cell-cell communication problem. But I think we want to bring that also into a topic modeling framework, which is the interested in the cell-cell communication. So why do we care about the interactions? Because interaction is important. So basically, especially in single cell, each single cell cannot exist by itself, and also it cannot be, in my opinion, it cannot be defined by itself. It can be defined in the context of interacting with the other cells. So a little bit of background story is basically it's heavily inspired by social network analysis. So in our case, because my PhD is more focusing on networks. So basically, social science, social networks has been it's been studied a long time. One example is how the political blogs actually citing each other, also co-authorship networks in some sort of field. And another type of network is like a football network, which is how different universities play against each other. So, and then co-authorship of the entire science sort of condensed matter in the archive papers. It's a heavily sort of complicated structures. But overall, in the social network studies, when people study the network, they usually first try out their method. They usually try out in the Zachary's Karate Club networks. It's really old network, and it's very small, only 34 nodes. But it's kind of epitomize what the problem is. So, and the social network is basically more or less they're friendship networks. So basically, each different vertex, different nodes are basically the different individuals in the Karate Club. I believe it was in Cornell. Yeah, something like that. And then we can look at the networks. So then we want to figure out some sort of groups from these networks. So then what's the our intuition is basically telling us that there's something somewhere in the middle. But is it true? Can we somehow formulate that in a mathematical way? So the people have been approaching this problem as a sort of like a min cut problem. So in the graph networks, what is the minimum cut that's cutting the minimum number of edges? So one trivial solution is basically just one min cut value 1 is just to take out some individual that's only connected to one person. then, but that's not what we want. Instead, what people have been doing is somehow identifying some influencing people. In this case, the person number 1 and the 33, 34. And then we kind of look at the source and the target sort of like max flow min cut problems. And then we can actually identify sort of like this type of min cut problems. So this is kind of the social network analysis that people have done. Can I actually present that as a sort of like a topic model-ish flavors? Yes, it's possible. One approach is a stochastic block model. So if you consider the network as a vertex by vertex sort of friendship networks, adjacency networks, and then you can have this A matrix that can be decomposed into 2 parts, which is Z, which is a membership for each gene, each person belong to one particular group, and then some sort of friendship strength, sort of parameter theta, and then a Z transpose theta times Z will reconstruct your adjacency matrix, which is great. But based on my experience, this stochastic block modeling is because we have 2 sides, Z on one side, on the Z on the other side, and that makes the inference a little bit more challenging compared to the other sort of topic modeling, NMF type of approach. But instead, if you're thinking about the problem from the edges perspective, which is all these edges, which vertex is it incident to? Is next connecting to? Then you can actually come up with some sort of edge incidence matrix, which is the vertex times edge matrix, and here the columns are basically the edges, and the rows are basically the vertices, so individuals. And then you can see it's at most only 2 incident vertices. This is useful, but if you if you plot that into A times A, then you can immediately see the product will give us some sort of group structures. So the edge group 1, edge group 2, you see some common patterns. Then if you do some sort of k-means clustering, then we can actually figure out the groups fairly easily. So in this karate club network, for example. So here is a cartoon example. I use this k-means clustering, assuming there are 5 clusters, and then we can actually sort of identify those edge groups. And knowing these edge groups, we can actually see what is the overlapping sort of membership groups of individuals that we can actually separate them out. Will this work well in the real biological data, biological networks? For example, I tested on the Reactome as a kind of another small version of the sort of toy example. So Reactome network is basically connecting the genes with each other if they're participating in the same sort of chemical reactions, biochemical reactions. And then it turned out that if you plot the edges and the genes, and then you can actually see the 15 clusters of the Reactome networks fairly easily. So this actually taught us a lot of things. First of all, so if you're thinking about this large networks from the perspective of gene or the vertexes, then it's probably hard. But if you're thinking about the problems from the edges perspective, so interactions, somehow interactions perspective, and then you can turn the problem into a simpler way. And another takeaway is you can immediately see that it's a more or less a topic model. Basically, you can see a large edge incidence matrix, and then you can sort of apply the same type of topic modeling. So we carry out the sort of idea to cell-cell interaction networks. So from looking at all the cell-cell interactions that we generated by some sort of similarity networks, and then we look at the source cell and the target cell, and say that the source cell I and target cell J, and then how do they interact? We can actually investigate their ligand and receptor protein, actually genes expressions, and the ligand I and the receptor J, so on. And then we look at these patterns as a sort of like gene expression patterns, and then we sort of identify the topics of the interactions. So in the cell-cell interaction, we're a little bit luckier compared to the other social network case because we know why cells are interacting with each other biologically. There is a source cell and a target cell, then there is a sort of ligand proteins expressed, and then the receptor proteins should be also expressed, and then they bind together and highly co-expressed together. Then it's more likely to have cells could have interactions. So we carry over that idea to cell-cell interaction networks and the topic model. Then the topic model is basically nothing more than we have some parameters on the ligand side and parameters on the receptor side, and there's some latent topics. So in this case, and then we can actually sort of trying to reconstruct the gene expressions with the one-layer model. So the whole model will look like this. It looks pretty complicated, but if you think about it, one side is just one topping model, and the other side is another topping model. So we kind of put them together and then combine them in the sort of latent space. So using this, we can actually decompose cell pairs into topics. So what's the benefit of doing that? It's because we can immediately see how what proteins in the ligand side, what proteins on the receptor side are they co-expressed together. So we can actually measure them like side by side. In cancer case, it turned out that it's very interesting because we can actually sort of broadly we can categorize all the cell-cell interactions into 7 categories in this particular data set. There is sort of seven interaction topics, which is different from the cell topic. Then we can actually see that maybe some of the cell types that we observed are actually the result of the interaction with the cancer cells. So, or the cancer cell, different types of cancer can be somehow defined by how do they interact with the surrounding environment. So that would be another takeaway from this modeling. In the end, we wanted to really make sure that it's really true signal. So we actually went back to the actual gene expressions and then see whether the sort of receptor proteins and the ligand proteins whether they are co-expressed together in a topic-specific manner. Then we kind of see that there's some nice modular patterns and they are sort of interacting with each other. So and then I propose that these 7 interaction topics are actually probably more informative than just the cell level sort of cancer type definitions. Because if you look at the cell types and then also interaction topics, and then you see some heterogeneity across different cell types, and then some of the cell types are it's mostly determined by cell types, but at the same time there's some heterogeneity across different cell types, so which actually teach us that there's some fine-tuning is happening, but that can happen as a result of the interaction between the cells. And the interaction topic from and the other side, we can actually look at different cell types. What are the interaction topics? How do they interact with the surrounding environment? So we can actually categorize them nicely. So and I hope that this could also teach us some sort of subtypes of breast cancer. For example, HER2 positive case. It's the same HER2 positive, but at the same time, there's some heterogeneity across the different interaction patterns. So ER positive as well and TNBC as well. So they're mostly interacting with the sort of cancer-associated fibroblast, but at the same time, there's some different patterns across different interaction topics. So then but the downside of this approach is it's heavily computationally expensive, because now if you were talking about the million cells, now we are talking about the million by million cell pairs of the data points. So now we are working on to make this approach more accessible to in a more scalable way. So, and another issue that I found was fundamentally our observation data, it's not suitable to answer all the gene-gene interactions, especially ligand receptor interactions. The reason is gene technically can be expressed in just one cell. So if you're thinking about cell I and cell J, so the same ligand receptor gene it can only present in one case. So which means there's a fundamentally missing values. So unless we actually fully perturb those expression patterns, there's no way we can actually really correlate that. So that's why enrichment pattern is useful in cell-state interaction analysis. So however, if you could somehow filling in those missing values, then we will be able to directly correlate this X and Y genes across different cell types, for example, then that will give us some nice correlations. That's hypothetically, that's what I'm thinking, but I'm not sure whether it's true. So there's some work to be done in this setting. So yeah, how much time do I have? Okay, so like 5 minutes is okay. Like 45 minutes. Yeah. Okay. So I will wrap up the talk. How do we speed up this topic modeling? So we show that the topic model is extremely useful. So it's very intuitive models, and we can directly interpret the results. So it's also useful in the cell-cell communication networks. But the problem is it's awfully slow, especially in this case. For example, I was fortunate enough to work on some of the large brain single cell data. So we have something like millions of cells in just one study, which is comparing to the whole human cell atlas. It's a huge data set, and the number of donors is huge, much larger than the entire database. I'm not sure about these days, like there was like so in 2 years ago. But say that you put together all the large single cell data, then there's some we're kind of hitting some numerical limits of the computer. So then because the number of elements is now going to be like a billion scale. So which is it's very easy to hit the sort of memory issue. So I think I didn't see memory issue in Python, but in R, yes, I saw that. Because R's integer type is not suitable to handle billion sort of indexes. So I'm not sure about this version, but it's R 3.6, but they could not do that. So even there's like something like a basic QC steps, it takes time. So like if you're dealing with the 2.4 million cells, so sometimes it takes about a day to sort of look at all the gene frequency patterns and all the densities or mitochondrial patterns or average gene expressions. So then it was pretty tough to do that. Then how do we actually speed up all this topic modeling, for example? So some people have some idea in the bioinformatics community. So this is one paper published 2 years ago in Genome Research. How do we handle a really large number of cells for single cell data? Their approach is, I think it's a pretty interesting. The idea is first they sort of identify some blocks, and then they somehow randomly project these high dimensional D genes onto some little d dimensions. And then they actually do the clustering on the little d dimension on this each blockwise, and then sort of combine the result and then take the majority voting type of approach. And then they take sort of average all these clustering results, and they ended up constructing sort of a full set large number of cells clustering results. At least in the time wise, it's very efficient. It can handle like 10 million cells in a real time, so minutes, in the order of minutes. At least in their simulation, it says it's a high performance compared to the other state-of-the-art clustering type of approaches in single cell. So yeah, it's a fewer computer resource, but as good as, or sometimes better, clustering results. The random projection idea is now, I think it's a kind of surfacing in many applications in single cell. So one thing is sort of random matrix factorization, random matrix multiplication to the single cell data has another benefit, because it can actually figure out what is the common signature versus some sporadic, unexpected signatures. This is their comments. The idea is basically, if you have a single cell data and then you multiply that with some random matrix, and then you look at the distribution of genes. And if the genes are somehow in the part of this complex system, then by random matrix theory, it will look sort of regular distribution. But if you were somehow outliers, somehow unexpected, then you have some weird shapes. That can be captured by just traditional random matrix theory, which is trace rhythm sort of the distributions, and then you can see some of the expected sort of eigenvalue distribution as opposed to something that unexpected. For this unexpected ones, we will probably think this is a kind of outlier genes or outlier cells, and then they show that it's very useful, so we can actually regularize the data well. So inspired by those papers, and then we sort of I asked Shishir to work on random projection sort of projects. I'm not sure whether it was the right decision, but we tried it out. And he came up with the acronym, which is ASAP. So annotating single cell approximate pseudobulk projection. So I think it's a good name. And then we multiply by some random matrix, like in this case, so type 8 random vectors with the 50K features, including non-coding genes. And then we can basically multiply this random matrix to all the 78,000 genes cells along the gene sort of 50K genes, and then we reduce the dimension to 50K to 8, which is great. And then we kind of combine those 8 vectors into some sort of like representative pseudobulk samples, 256. You can guess how I came up with the 256 by 8. So then it turned out that it works really well. And then look at the 256 pseudobulk samples, random pseudobulk samples, and then match the known cell type annotations. It turned out it's so diagonal patterns are so satisfying to me. So it's a colored version is much easier to visualize. So different sort of brain cell types, for example, in the cortical regions, and then you kind of enrich this random known cell types in the random samples along with that. So if you compare with the marker genes of the brain cell types, and then you can see so marker genes are somehow nicely coming along in terms of gene expression patterns. So it's nice. However, pseudobulk sample is it's not that simple. You see some sort of some topics there. So suppose we do the NMF perform on the random projected pseudobulk data, and then you can see some topic proportion patterns, which is not very intuitive to me, but turned out that it's very informative if you do somehow a cell clustering based on that random projected NMF result. Then you can see some nice patterns of the t-SNE and UMAP, and in both cases, it can nicely separate the known cell types into sort of independent components. So here's a different try of the same analysis. Then in the end, we were wondering whether we can use the same approach, mixing, for example, 2 different studies. In this case, mixing with the glioblastoma and then the LM Brain Atlas cell type annotation data. But it turned out that it's not that positive. So you see huge batch effect. So glioblastoma data is somehow separated out from the normal brain cells. Maybe it's true. So I wanted to make sure that it's really true. But it turned out that no, it's a really batch effect. The method doesn't know how to handle the batch effect well. So Alzheimer's disease data, so with the LM Brain Atlas, so it turned out that it's a different data set. We're kind of different. So using that, it's a still promising result. I think it's just the ongoing work, but I think it's our effort is to make the sort of topic modeling go faster. So that's how I do that, how we are up to. Yeah, so for that, I will just end the talk because for the interest of time. So today I discussed some updates from our lab, and then we look at the probabilistic topic models, and then cell-cell communication network analysis can be done by topic modeling as well. And then Shishir is also working on another idea to make this really scalable across, I don't know, millions of cells or 10 millions of cells, tens of millions of cells. Then using this, I'm hoping that this is a basic ingredient that we can actually do interpret the biological mechanisms in the single cell data in a more interpretable way. So yeah. So without further ado, yeah, thank you so much.
We have time for a few questions that Dr. Park would like to answer.
Yeah, doesn't have to be single cell. Any questions? Yes.
So in the by the way, it was amazing.
Thank you.
In the last part of your talk, you mentioned about the batch effect. So I am assuming this because of the biological model that you are using based on. So you are using the genes as your indicator of the of your models of it. But like when you compare like brain data, brain cells versus the other tissues,
Right.
organ type, their expression itself they are so different.
Right. I agree. Yeah.
So maybe you can integrate some other way, like what can be.
Yeah, that's the direction that we are sort of thinking about. Then we should somehow select the genes. We have to somehow categorize genes into two categories. One is sort of ubiquitous across different maybe tissues or organs, or some genes are somehow really tissue or organ specific. So
like
yeah.
In the end, like you are already like in this gene level.
Right.
But like, how do they like regulate?
How do they regulate?
Yeah.
Oh,
like in between organs.
Oh, I think I think of the gene expression as something like close to the steady equilibrium state, because that's the state that defines gene expression state that defines the identity of the cell types.
Like in the like after the genes are transcribed, there is a post-transcriptional modification.
Right. That's a whole different different level of complexity. Yes. I would in that case hopefully a collaboration with some groups here. We are interested in looking at the sort of post-transcription regulation and the post-translational regulations, all kinds of things. So we need to model them as another layer of topic model. Yeah.
Now you would just count them to the same gene, right? Different splice forms and things like that.
Right. Exactly. Yeah. If you have a data set, let us know. Yeah.
Do you have any other questions?
Okay. No Zoom. Okay. So, but yeah, feel free to reach out to me anytime you have cool ideas, crazy ideas. I can try out. We can try out. She sure can try out.
Okay. So I guess that will conclude our talk. Thank you so much, Dr.
Thank you.
Park.
Yeah.
Yes.
Thank you.
Again.